Hello, everyone, and welcome to this podcast for cell biology. In this podcast, we're going to continue our discussion of the cell cycle. If you remember, on the last podcast, we talked about the cell cycle. And we talked about G1, S phase, G2, and M phase. We also talked about the G1S checkpoint, the G2M checkpoint, and then the spindle checkpoint during mitosis. And remember, we talked about cyclin-dependent kinases and how they were inactive until they were bound to cyclins. And then once they were bound, they were still inactive until they were activated by a phosphatase. Not active, and then active. And this allowed the cell to build up at a high concentration all these cyclins and their CDK counterparts, but by having them be finally activated by phosphatase, it allowed them to be quickly activated at the exact same time, which allowed for that very abrupt on-off switch. Now, what we didn't do was spend too much time talking about this activated cyclin-dependent kinase cyclin component here. What does it do once it's activated? How does it drive the cell cycle? And that's what we want to spend some time talking about in this podcast. So at the end of M phase, let's just remind ourselves that a new cell is going to be produced. And I'm going to just give it 2 chromosomes here. During M phase, there are several CDK cyclins that are present. Some of them are active, some of them aren't active. Some of them are inactive and are existing from the previous steps of the cell cycle. But some of them are active. Now, if this cell entered G1 and it still had all these CDK cyclins in there, there would be nothing to prevent it from jumping into the next cell cycle without regulation. So it's important that when this cell is produced, that it comes up to this checkpoint and stops. So the only way we can assure that it's going to do that is by getting rid of the CDK cyclins. So to accomplish this, CDK cyclins become inactive towards the end of M phase. Inactivating these CDK cyclins is accomplished by inhibiting formation of new cyclins. There'll still be some CDKs around, but they inhibit the formation of cyclins. And by doing so, you can't activate the CDKs. Cyclins are also degraded. And finally, CDK inhibitors bind to and inhibit CDK cyclins. By using multiple strategies, we generate a very robust pathway to inactivate CDKs. By doing this effectively resets the cell cycle. So in the M phase, we get rid of all the cyclins and CDKs, or at least make them inactive. The new cell doesn't have any active CDK cyclins. Therefore, when it hits this cell cycle stop signal, it stops. It doesn't keep going. At this point here, it will assess the environment and determine whether or not it needs to make a new cell. If it does make a new cell, the first step will be to enter into S phase, where these chromosomes will be copied. So let's talk about what happens at this point here with this resetted cell cycle, what's going to drive it into S phase. So what we have then are these 2 cells that are stopped at the G1S checkpoint. They both have these receptors on them, and these receptors will bind to chemicals secreted by other cells called mitogens. Mitogens are extracellular signaling molecules that stimulate division, cell division. So if no mitogen binds to it, it will stay at the G1S stop. It could stay there for a few minutes, a few hours, a few days, forever. If it stays forever, it will enter into that G0 stage. However, if the mitogen is present, the cell is receiving a signal from neighboring cells that it needs to divide. And so when the mitogen binds here, we'll have an intracellular signaling pathway that will take the G1 cell into an S phase. So mitogen binds and it will enter S phase. But how does that drive the cell cycle? How does it push it forward? To help this switch occur in a more rapid way, before the mitogen binds and during G1, G1S cyclins and CDKs, as well as S cyclins and CDKs, will accumulate. They're not active yet, but they will accumulate. And as they accumulate, they'll be poised so that they can rapidly jump into the S phase once that mitogen binds. So let's talk a little bit more about what the cyclin CDK is going to do to activate cell division once the mitogen binds. So let's draw a fairly large cell here so I have some room to work with. And I'm also going to draw a disproportionately large nucleus here. We'll have a receptor here, and there's probably several receptors that will bind the same mitogen. So the mitogen binds here through various steps and pathways. An intracellular signal is produced, and it will enter into the nucleus. That is, one of the final targeted proteins will now enter the nucleus. Now remember, in the nucleus, we have our CDK here, and it is specifically, in this case, a G1S CDK, or it could be an S CDK. And we have the cyclin bound to it. And remember, it's inactive at this point, so it has that phosphate group added to it. This protein here will remove that phosphate group, and now we'll have the activated CDK cyclin. So now it's activated. Now once activated, it being a kinase, it's going to phosphorylate a target that's going to drive the cell cycle into S phase. So now we need to introduce two more proteins here. And that is RB, which stands for retinoblastoma, because it was first identified in the eye tumor called retinoblastoma. And it's bound to E2F. E2F is a transcription factor. But as long as E2F is bound to RB, it is inactivated. So when you know it, our CDK cyclin, our S CDK cyclin, will come along here, and it will add a phosphate group to the retinoblastoma protein. Upon binding this phosphate group, the retinoblastoma protein will have a conformational change, causing E2F to separate from the retinoblastoma protein. And once it's separated, E2F will bind to specific promoters, to specific genes here, and drive the transcription of these genes. And these genes are genes that play a role in DNA synthesis. So to review, mitogens bind, there's a signal produced. The final activated protein from this signal will remove the phosphate group from the CDK cyclin. Now the CDK cyclin is activated. Once activated, it adds a phosphate group to retinoblastoma. Retinoblastoma protein goes through a conformational change and is removed. E2F is now activated. Let me write activated here. And once activated, it will bind to the promoter regions of genes that need to be turned on. And these genes have specific roles in DNA synthesis. So up to this point here, the cell is stopped. The cell receives these signals that say we need to make more cells. And through this long pathway I just described, the cell will immediately jump into S phase and begin copying the DNA, as we see here. Now, I want to talk about what happens if we want to stop this and make it so it doesn't go forward. And one of the ways that it happens is if there is DNA damage. So if the cell detects DNA damage, signal or no signal, it's going to stop the cell cycle. So how does it stop the cell cycle even in the presence of a mitogen? Because the last thing you want to do is copy the DNA if there's a mutation in it. I want to keep all this here, so I'm going to just extend our cell, make it even more large or larger, and make the nucleus also larger. So remember, in this particular situation, there's been a signal for the cell to divide. So the signal has been generated. This protein should be dephosphorylating this to make it active. But there's a DNA damage that needs to be fixed. So let's draw some DNA up here. And I'm going to just make a couple asterisks on it, indicating that there's mutations in this DNA. There's this protein, very famous protein, called P53. P53 detects this DNA damage, and through another process that we're not going to talk about, it becomes an active P53. So I'm going to put an asterisk there just to indicate that it is now active. Being active, P53 will activate the transcription of another gene and make it active called P21. And what P21 will do is it will block the activation of CDK cyclin. DNA's damage is detected, P53 becomes activated. Once P53 is activated, it activates the transcription of P21. Active P21 then will stop the cell cycle by inhibiting CDK cyclin from becoming activated. As long as this is happening, CDK cannot add a phosphate group to this retinoblastoma, so there's no conformational change, and there's no activated E2F. So E2F cannot activate the transcription of the genes necessary to go through the cell cycle. It will remain bound. P53 will also help in the process of the cell to decide whether to go through apoptosis, because if there's too much damage, the cell will just go through apoptosis, or it will start a set of pathways to repair mutations. So again, the cell is saying, the cell is receiving a signal to go past this cell cycle checkpoint, but P53 detects a mutation. P53 through P21 will block this enzyme until the cell repairs the DNA or the cell goes through apoptosis. If it repairs the DNA, once the DNA is repaired, P21 will be degraded. It will remove its inhibition of CDK cyclin, and then once again, retinoblastoma can be phosphorylated so that E2F can activate the transcription of these genes. So now we're at the point here, by transcribing these genes, the cell is firmly established in S phase. And now let's pick up from here and ask what's happening during S phase so that the DNA can be faithfully copied. So during S phase, what happens during S phase? The DNA, all the DNA, must be copied quickly, but also faithfully. And we'll talk in a little bit how this protein called SCDK helps drive this through the S phase. So the first thing that has to happen is that the DNA has to be prepped and ready for replication. So here's our chromosome here. And throughout any given eukaryotic chromosome, there will be several of these sites that are called origins of replication. This is a site that will recruit a large complex of proteins called the pre-replication complex. And there are several of these, so the pre-replication complex has to bind to each of these origins of replication. And in fact, it will do this during G1. It'll form this complex, and I'll talk about what's in this complex in just a moment, but it'll form this complex in G1 so that once it's been triggered to enter S phase, it can immediately start copying the DNA. Again, that very abrupt on and off switch is possible because of this proper planning during the previous stage or phase. So what is this pre-replication complex? Well, let's talk about some of the proteins. And the first one that binds is called ORC. The ORC stands for Origin of Replication Complex. It's the first one, and it's important because it identifies where these origins of replication are. That's its job. Once it's identified where these origins of replication are, it will begin to recruit several other proteins. The next one that comes in here is called CDC6. This will help signal the start of replication in just a moment, but for it to do that, it brings in this third protein. And we'll put it right here, and it's called helicase. Helicase is important because as the DNA begins to get replicated, it will open up the double helix. And it does so by breaking those hydrogen bonds that are holding the double helix together. So at this point here, and there are other proteins here, I'm just not indicating them all, I just want you to know these three. At this point, it is poised and ready to replicate the DNA. It is just waiting for that signal that it's time to move past this checkpoint, meaning there's no mutations in there, there's been a mitogen that has bound to the receptor on the outside of the cell, it's ready to copy our DNA. So once, if you remember from the previous whiteboard, once E2F starts transcribing those genes, some of the genes it transcribes are genes that will activate SCDK. So let me draw that out here. E2F will transcribe genes to activate SCDK cyclin. So we have the activated SCDK cyclin. And this being activated, what it will do, it will add a phosphate group to helicase. This will allow helicase to start doing its job. I'm going to erase this to kind of show you this next stage. The helicase will begin to unwind the DNA, so it kind of loops out like so. So we'll draw the helicase here, breaking those hydrogen bonds. And as it's breaking those hydrogen bonds, other replication enzymes come into the complex and begin to copy the DNA. So this helicase will begin to move down here, open up the DNA, replication enzymes come in here and copy the DNA. And that's because the activated cyclin-dependent kinase adds a phosphate to the helicase. Now, activated CDK cyclin also does one other thing. Well, it does a lot of other things, but one other thing I want you to know. It will add a phosphate group to CDC6. This will trigger CD6 to fall off of this complex and become degraded. The reason this is important is that if we didn't remove CDC6, the DNA would be replicated over and over and over again. So it's really important to remove this CDC6 so that the DNA is only copied one time. If this was prevented from happening, as I just said, the DNA would copy itself multiple times. Activated SCDK cyclin phosphorylates helicase and CDC6 so that the DNA is copied once and only once. Okay, so we're moving right along here. The DNA has been copied. It's in G2 here where we have our replicated DNA, but it needs to stop here at the G2M checkpoint. And remember, it has to stop here because it needs to make sure that all the DNA has been copied, there are no mutations, and if there are mutations, they've been fixed. And then it can go past this G2M checkpoint. Also at this checkpoint, several enzymes are being produced and activated so that it can move into M phase when it's given the signal to go past this checkpoint. So let's go into M phase. What happens during M phase is our very good friend from way back when called mitosis. During mitosis, the copied DNA is segregated into two new cells. So we'll have our cell here, and we have our two chromosomes that have been copied. So essentially what we're looking at are two red chromosomes and two blue chromosomes. Each of these red ones are identical genetically, same sequence that is. But what we want to end up with are these two cells like this, one of each. So the process that's going to happen, and this will be in a separate podcast, but I'm just going to write it here real quick, is these chromosomes will line up along this middle of the cell. Our centrosomes that we've talked about previously will be copied. They will send out spindles to attach to each of these, and then they will break this connection, and we'll talk about that in a moment, what this connection is, and pull to each cell a blue copy and a red copy. Blue, red, blue, red. But how does the cell know when to do this? And I know you will probably be very shocked to hear that it's all going to be triggered by this enzyme called MCDK. I hope you were sitting down when you heard that news. So what does MCDK do? It will prepare DNA for segregation, and it will also induce the duplication of the centrosome. These structures here. Prior to this, they just exist as a single structure, but then they get copied. And MCDK will do that. So what will happen is we'll have our inactive CDK, MCDK, and it will be bound to the cyclin. So this is inactive. It becomes active by being phosphorylated. So I should put some phosphate groups here. So it gets dephosphorylated by CDC25. CDC25 is activated when it's been phosphorylated, and I'll talk in a moment how it gets phosphorylated. So CDC25 is a phosphatase, and it will remove these phosphate groups, activating our MCDK. CDC25 will remove those phosphate groups, and now we have an active MCDK. The active MCDK does a couple things. The first thing it does is it's involved in a positive feedback mechanism. I'm sorry this is kind of merging with this drawing. I thought I would leave this here just in case we want to reference it, but it's kind of making it a little messy here, so I'm sorry about that. But anyhow, this active MCDK plays a role in phosphorylating inactive CDC25. So this is how active MCDK is responsible for the continuation of activating all of the inactive MCDK. It's an effective mechanism to provide this kind of quick switch to produce a lot of this active MCDK so it can go on and do its next job. So the other thing active MCDK does is it will make this chromosome condense. So within a cell that's in G2, this DNA is not visible with a microscope. It's visible with a dry erase marker, but not with a microscope. And so it's just kind of really stringy in here. But what will happen is, as the cell enters M phase, these chromosomes condense. We never draw them condensed when we draw these figures here, because this is just to show you academically what's happening. But in the actual cell, they condense to a very tightly structure and a visible structure under the microscope. And this is caused by a protein called condensin. Condensin is inactive in G2, but in M phase, MCDK will add a phosphate group to it. Adding the phosphate group to it, it's able to facilitate the condensation of these chromosomes. So active MCDK does several things. Two of them that I want you to know is it provides a role in continuously rejuvenating more and more active MCDK. And then another thing it does is that it helps condense the DNA into these nicely condensed structures by phosphorylating condensin. So now I want to come back to this drawing here and talk about how this sister chromatid and this sister chromatid, these identical pieces of DNA, are held together. So these two blue ones and these two red ones. They're held together because of a protein that I'm going to show here in brown, and this protein is called cohesin. It actually forms during S phase, and its job is to hold these pieces of DNA together. But it's at M phase, through the activity of the cyclin-dependent kinases, that will degrade cohesin at the right time, so that during anaphase and telophase, they will pull these chromatids in opposite directions to give us these two here. If this doesn't happen, we can end up with cells that have more or less chromosomes. Having more or less chromosomes is a problem, and that leads to various conditions that you'll learn about later, not in this class, but in genetics or perhaps in other courses, called like conditions like Down syndrome. So these cohesin molecules are degraded, which allows the efficient separation at the right time of the chromosome. And at this point here, we've ended the cell cycle in all of its glory, where we've produced two new cells, and they can each enter into their own cell cycle. But remember, these two new cells that have just been generated have also been reset, removing all of the active CDK cyclins. Okay, that's all I have for this podcast on what happens during these different stages of the cell cycle. There's one more podcast. It's on mitosis, what's happening specifically to the chromosomes during M phase. But that's it for this podcast. If you have any questions at all, let me know. If not, I'll see you in class. Bye.